Now we're going to get a little more complicated. So far we've looked at simple dominance versus recessiveness, where one trait can mask the expression of the other. But there are traits that are not either or so definitively. We're going to look beyond simple inheritance patterns. First, incomplete dominance. Just like it sounds, this happens when a trait is incompletely dominant. In other words, the heterozygote has an intermediate phenotype between the two homozygotes. Looking at curly, straight, and wavy hair is a good example of this. If we have someone who has naturally curly hair and reproduces with someone who has naturally straight hair, theoretically all their children will have wavy hair. If we have two wavy-haired persons that reproduce, then we get the expected phenotype of a 1 to 2 to 1, as in one curly hair, two wavy hair, one straight hair, possibility for each time a child is produced. Let's look at that in more detail. So if we have a person with straight hair represented as this H1, and a person with naturally curly hair represented as H2, notice here that we have alleles that have the same letter. They're both dominant in their own right, and so that's why we have the subscript. Wavy hair is going to be represented by this H1. So if you have two wavy-haired people that are reproducing, that's represented by this Punnett square here. So this H1 coming here. So that represents a straight-haired person in this first square. Likewise, we get two wavy-haired possibilities in these two squares. Finally, we get the possibility of a curly-haired person in this final square. This is where we get this phenotypic ratio shown here. If we're looking at the concept of codominance, that is when alleles are equally expressed in a heterozygote. For example, with the blood type AB, red blood cells have both type A and type B surface antigens that are possible. We can assume that both alleles code for a product, and we observe the result of both products being present. We'll look more into blood types as we go along. Multiple allele inheritance is when a trait is controlled by multiple alleles, or more than two. And so the gene exists in several allelic forms. So far we've seen an either or. Now we're going to look at more than two. But keeping in mind that each person for a diploid organism is only going to inherit two of the possible alleles. So with the ABO blood type group, there are three alleles that are responsible. Here are the three alleles. We have IA, IB, and little I. This first allele, the IA, encodes for A antigens on red blood cells. IB encodes for B antigens on red blood cells. So with these two alleles, we have that codominance that we just looked at, since they both encode for a product. However, for this little I, since it's recessive, it does not code for A nor B antigens on red blood cells. So we have this both IA and IB are dominant over little I, and like we just said, they're codominant. So the phenotype, you could have either one of these four blood types, type A, type B, type AB, or type O. The genotypes are as follows. So you could be homozygous dominant for type A, or you could be heterozygous. Similarly, you could be homozygous dominant for type B or heterozygous. To have type AB blood, you're going to have IA, IB, and in order to have type O blood, you're homozygous recessive. So again, both IA and IB are dominant over that little I, and they're codominant. Now the Rh factor is inherited separately from the ABO blood types, An Rh positive person has the Rh antigen on their red blood cells, and there are multiple recessive alleles for Rh negative phenotype, all recessive to the Rh positive allele. But first we're just going to look at the ABO blood type, not whether it's positive or negative. So an example of how to work a blood type Punnett square. Again, even though we have multiple allele inheritance here, each individual only has 2 alleles for that trait. So if you have parents of type B and type A blood, but they're both heterozygotes, we can theoretically get offspring with any blood type looking at phenotypes, because we could get an offspring with type AB, could get one with type B, type A, or type O. So there can be some surprising results with blood types. Polygenic inheritance occurs when a trait is governed by 2 or more genes or sets of alleles, and not just a single set as we've seen earlier. Dominant alleles have a quantitative effect on the phenotype. That means they add up, or that these effects are additive. The result is we have continuous variation of phenotypes. So skin color is an example, because we don't have just either or, right? There's a lot of different skin tones. Eye color is another example of this too, since there's at least 7 different pairs of alleles that govern eye color. Looking at skin color, which is one example of polygenic inheritance, it's the result of pigmentation produced by melanocyte cells. There are over 100 different genes that influence skin color. For simplicity's sake, we're just going to look at 3. So if we use 3 pairs of alleles as a simplified example, we have A's, B's, and C's. Each dominant allele or capital letter is going to contribute to pigmentation. When a very dark person reproduces with a very light person, the children will have medium brown skin. When 2 individuals with the genotype big A, big B, big C, little c reproduce with one another, their offspring may range in skin color from very dark to very light. Here, if you were carrying that out, the phenotype distribution follows a bell-shaped curve, which is characteristic of a polygenic trait. And so this implies that few individuals have the extreme phenotypes. That would be being very, very dark or very, very light. Instead, most have the phenotype that lies somewhere in the middle. Looking at possible genotypes from this cross, we could have anywhere from no pigmentation or being very, very light, looking to different shades of being light, medium brown, into dark and very dark. And as we can see, we have this bell-shaped curve where more of a worldwide population is going to lie somewhere in the middle here, and we have less extremes on either end. One of our final concepts to look at whenever we're looking beyond simple inheritance patterns is that of environmental influences. We mentioned earlier the concept of nature versus nurture. So just how much does environment have an effect on genetic expression? Environmental factors such as nutrition or temperature can influence the expression of genetic traits. If we're looking even at skin tone, if we're looking at how much UV exposure somebody gets, that's going to affect whether more melanin is produced by our skin or not. So polygenic traits are especially influenced by environment. We have height that's influenced by nutrition. The primrose is influenced by temperature. Plants have white flowers when they're grown above 32 degrees Celsius, and plants have red flowers when grown at 24 degrees Celsius. Looking at the case of height, we have differences in nutrition that are one of the factors that bring about this bell-shaped curve. Represented here, we have males in the dark and females in the light. But overall, we have fewer that are at the extreme ends that are especially tall or especially short. Most lie somewhere in the middle. If we're looking at populations over time, we can see that overall height has increased, largely due to better nutrition being available. Another example of environmental influence is looking at Siamese cats and Himalayan rabbits. They're darker in color on the ears, nose, paws, and tail. The Himalayan rabbit are known to be homozygous for the CH allele involved in producing melanin. That's the same pigment that's responsible for pigmentation of our skin and eyes. The enzyme coded for this gene is active only at low temperatures, and that explains why the black color is limited to the extremities, where body heat is lost to the environment, as in the core temperature is going to be a few degrees higher than those in the ears, nose, paws, and tail. So if we're looking at a Himalayan rabbit that naturally has darker paws, darker nose, darker ears, can't see the tail, but that would also be black. I wouldn't recommend really carrying this experiment out at home, if you were to shave your Himalayan rabbit and put an ice pack over that area, then that fur should grow back dark. So showing how temperature can affect phenotypes of plants and animals.